Pardon.
Ses böyle geliyor.
Ben de tavsiye ederim ama. Tavsiye gelmedim. Tekte ben.
Muhammed içeride. Bu konuyu düşünün ya. Gerçekten.
Kim online bağlı? Online bağlanan arkadaşlar bir sinyal verebilirler mi? Beni duyuyorlar mı?
Duyuyoruz.
Beni duyuyor musunuz arkadaşlar?
Duyuyoruz hocam.
Ha, harika, tamamdır. Hocam başladın mı? Evet. Ben onları buraya gönderirim, merak etmeyin. Okay, thank you for all coming. Today, in the Institute seminar, we have one of our faculty members who is very active in medical imaging. Focusing more, we have joint project on nuclear imaging, also instrumentation. It's a great pleasure to have him here, Professor Kremich, who will talk about precision imaging for non-invasive precision medicine of cancer. With a focus, we know he has several projects for the past maybe 10 years, right? With several graduates already on master's and PhD program, have done thesis as well as several important publications. So it will be a great pleasure to hear him speak. Thank you. The floor is yours. I can close it if you want.
Yeah, thank you very much. I don't have my glasses. I forgot my glasses. So. Okay. Now I see the screen.
If you speak a little bit more towards the camera, they will.
That's where the microphone is.
That's why the microphone.
Yeah, okay. Is it okay now? Is someone checking it or?
Yeah, they will complain if they don't hear it.
There are online people as well.
There's no complaint. So, Mohammed, you finished eating or?
No, I think
We're still eating? Okay, hi everyone. We'll talk about precision in medicine in general and then also in imaging. But in general, we have a precision issue in medicine. We'll talk about this. And then we'll focus on molecular imaging and how we can improve therapy for cancer and also other diseases. By doing molecular imaging, but using precision medicine and also precision in instrumentation. Now we are a bit different than other groups. We use precision in two meanings. Precision medicine means, I will show you what it means now, but it means in the world right now, what they mean by precision medicine is using genetic data and other types of data to give individualized therapy. That's fun. But we also use precision in the sense that we want to use precision instrumentation because we still have issues with instrumentation in medicine, including imaging systems. So when we combine both, we hope to get like much better accuracy and efficiency and efficacy in therapies and also in diagnosis. So that's what this is about. Actually, here we have two different presentations. One that I gave in Spain, in Granada, on more like instrumentation, and the second one is the one that I gave for the European University of Brain and Technology, which is more on precision imaging and precision medicine. So that's about it. Okay, so when we look at precision, the definition of precision, we take two definitions instead of one. So the first one is precision medicine is an innovative approach that uses information about an individual's genomic and other clinical environment and lifestyle information to guide decisions related to their medical management. So this is fairly new. It was the President Obama that started this project a few years ago. And it has some actually many followers, and today medicine is going towards precision medicine. So instead of giving one medication for all cancer patients, now we try to individualize treatment according to the lots of data that we collect from the patient, especially genetic data.
Precision.
That's new, and actually there's a lot of progress going in this direction. The second one is more historically related to the
Precision.
institute. But what is it? Our institute started with the idea of improving clinical engineering, especially in Turkey, but also regionally, with a UNESCO project, and also with ECRI, which is an institute in Philadelphia, United States. So we started this project with the aim of improving the quality of medical services, and of course, the medical technology. So in this case, we're talking about improving the precision or precise instrument, which is a tool that can be controlled very accurately, so it produces very accurate results. Today, it's not the case. Like, for example, we have radiotherapy systems that unfortunately do not target the tumor very well, or we have imaging systems that cannot really image precisely the tumor, etc. So we have some issues with instrumentation. So we try to address both issues at the same time to improve both diagnostic accuracy and therapeutic accuracy. So, so we'll see some of the issues. But before I get into the issues, let me say that we are collaborating with a number of institutions. We're collaborating with ECRI in Philadelphia, JCHO in Chicago, which is the accreditation, and we've done some surveying of hospitals for the quality. We also work with WHO, which is out here. We work with Drexel University. We also work with Moffitt in Florida, and with all the gentlemen that you see. And now the precision medicine medical technology, let's start with this first, and then we'll switch to precision medicine, which is the second part. So the first part, let's see some figures. We see, for example, only 50% of 1 centimeter lesion can be detected in X-ray film. So we have a number of numbers here that show us that we really lack precision that we have in other industries. Like you can take other industries such as aviation. We have lots of precision, we have accuracy. You don't see planes crashing, right? So, but here in medicine, we see 50%. That's a pretty low number. We have 30 to 70% of breast cancer detected at follow-up mammography, retrospectively seen on first mammograms, interpreted as showing normal findings. So we see we have really a high figure for inefficiency and errors. So up to 40% missed fractures in emergency rooms. Inefficacy of drugs, like today in cancer drugs, they have 25% efficacy for a drug, which is very low. So, and also too much trial and error, especially in cancer therapy. Today we see that you try this drug, it doesn't work, so you try another one, and then the patient dies. So, so we need to be more precise. That is, give the right medication at the right time to the right patient. We're not doing this now. We're just giving the same drug to everyone, and then some people benefit and some people don't benefit, which is not suitable anymore. So we have all these problems to solve. But when you look at other industries, such as electronics industry, this Six Sigma methodology has been started in the electronic industry by Motorola in 1985. So we're talking about a long time ago, almost 40 years ago. And that means you need to have 99.99966% of the products manufactured. They need to be okay. That is 3.4 defects per million is acceptable according to Six Sigma. But in medicine, we're talking about inefficiencies or error rates of 20%, 30%, 50%. So, look at the difference in these numbers. So that means we need some quality improvements, and some of the methods that we use in quality improvement to apply them in medicine. So, what are some of the medical technology risks and hazards? We have a number of issues, such as unnecessary radiation load. You go to a dentist, okay, there is, for example, something pretty simple, and then you end up with thyroid cancer. You may. It's not 100%, but you have a certain percentage of patients who get thyroid cancer because of X-ray. Now, this should not be acceptable. Inaccuracies in radiotherapy. Yeah, because the tumor is moving and it's not well contoured, and all these things make it that you may not be very accurate. Undetected nodules in imaging, or electromagnetic interference, or electric shocks during catheterization, for example. So all these issues are still a problem in hospitals. And so what we do is we use statistical design techniques, which will optimize performance, reduce variation. Variation is our enemy. We don't want variation. Like variation means one patient will get well, the other one will not get well. That's variation. All of them should be well, right? So variation is not where we are trying to fight this. Robustness against uncontrollable variation. So there are always uncontrollable variations. Like if it's a car, then you know that it may rain, it may snow, and your car should still be able to go. So I've been trying to buy a car in the United States, a big car. I wanted a big car, like everybody. And then I tried on snow, it didn't work. So I gave up on my dream, and I had to go with Volkswagen, a small Volkswagen, which works on snow, but it's small. Okay, I was a bit sad about this one. So you need robustness against uncontrollable variations, and those can be components that vary, patients that vary some external conditions, and tolerance is that design comes after. So we have all these different tools we can use for designing new medical technology. And the goal is obviously we want 100% efficiency or efficacy, or maybe something close, low cost, no side effect, and predictive biomarkers. Predictive biomarkers means you need some biomarkers that will predict if that particular therapy will work for a particular patient. This is something we're working on. So if we have this predictive biomarkers, then we do not give a medication to someone and it would not work. That should not happen. Because, cancer patients don't have too much time. And, if they lose like 3 or 4 months for medication, with a medication that doesn't work, it's actually, a pretty bad situation. So we need predictive biomarkers, some biomarkers that will tell which patient will benefit from which therapy. So, okay, here is a slide that shows the old way of quality and the new way of quality management. In the old days, people used to manufacture, and anything that didn't conform, they used to throw it away or maybe repair, but that's too costly. And today the idea is to change the design so that actually, you know, when you change the design, that Gaussian shifts to the left and there is no defect. So basically, there's an inherent quality in the product. And we should do the same with medicine. Now, when you want to improve the quality, you have a number of tools you can use, like use standards. At least you reduce the variation. You can use quality indicators or technical specs, or everything becomes quantitative. You can do quality improvement techniques. We have some techniques there. Design methodologies such as Taguchi or response surface methodology, which we use in our work, and also robust design, which also is a work by Taguchi, a certain guy from Japan. So if you look at standards in medicine, we have many standards like JCIEK, the ISO, professional associations, ACI, SNM, NEMA, and so on, many others. So one of the easiest things to improve something to make sure the quality improves is to use a standard. At least it's not optimal, maybe, but at least it will make things uniform and reduce variations. And also, when you quantify things, you reduce the errors. Like you can check anesthesia complications rate, X-ray repeat rate, C-section rate, or if it's a camera, the resolution of the camera, in front of the camera, all these technical specs which are quantitative. When you measure, then you reduce the quality. You cannot have quality if you don't measure something. It's not good enough to say we have good, we have good, a good hospital or we have a good hospital. Okay, so I'm going to, and then at the end of all the quality improvement, you have a statistical process control, which we use to track the quality, and not only the quality of a technology or a process, but also the patient. So we track patients with SPC to make sure that everything is under control. So that's the tool that we use. And there's a PhD student now who's going to use SPC for asthma monitoring. You can see an example here to make sure that there is no asthma attack. And what we can do is we can actually predict if an attack is coming from this graph statistically. So there's ways to predict, it's like predicting earthquake, but here we predict that there will be some problem with the patient before it happens. So we can actually warn the patient something is wrong. Maybe there is some dust coming up, you need to take action. Maybe this your medication. Taguchi is a guy who developed a number of methodologies for this a good design, and response surface method is one of them that we have used. I'm going to show you some examples. I'm not going to get into the procedure, but we have used this procedure to improve some really seriously erroneous processes. And this can be used in many examples, in many areas such as electronics or transducer design or hemodialysis, PCI imaging systems, and so on. Here's an example that was used. This is not our work, but it was done to design a finger probe. And they used this Taguchi method to improve the accuracy of the system. if there are some numbers which, for example, the standard deviation, which is the variation, has been reduced from 1.18 to 0.85 by applying this method. So we need to apply this method. And now let's go to nuclear medicine and PET and Spec and gamma camera and molecular imaging, which we will talk about, which basically is in vivo biology. This PET and Spec can show biology in vivo, which is different than CT or MR. So you can see the biology in vivo and in real time.
Functional imaging.
And this oncology, in oncology, you have many applications, such as here is FAT, fluorodeoxyglucose, where you can see a tumor, and you see the arrows. The reason why you can see the tumor is because the tumor can absorb glucose to a higher rate, like 8 to 10 times more than the background, so you can see the tumor. All right, so here is an example of a good imaging system and a bad imaging system. Here you can see the two masses in the breast, and on the right you cannot see them. So it's important to optimize them. So there was, up until now, optimization method was lacking in this system. So we have developed we used some optimization methods that were not used before, because in the past, it was one variable at a time, and now we have a simultaneous way of improving and optimizing systems. So that we published in a Transactions Journal of Nuclear Science. And we use contrast noise ratio and other model observers for quantifying image quality. So in our work, I'll show you some examples later on. This is Monte Carlo simulation. We also use Monte Carlo simulation, the statistical simulation, to simulate an imaging system. So here is an example, improving the detectability of overactive glands in dual-phase parathyroid Spec CT. Here again, we show that by selecting the right parameters, protocols, you can significantly improve the visibility or detectability of a beam. So you can see that. This is from a paper in biomedical physics and engineering. And we use response surface method, which is used to optimize. You can see that these are some contours, and if you go in a certain direction, you get the optimal value for the design parameters. Okay, here comes now precision medicine. So far, we talked about general precision in medicine, the problems of errors and all that stuff. But now let's talk about precision medicine, which means something a bit different, which means that we are looking at a whole lot of data on a patient, including genetic data, to find out which medication will work for that particular patient. For that, I use an analogy. If you try to open the door when it's dark, obviously you're not going to see the key, and you're going to spend some time trying to find the lock and open the door. But if you have enough light, then it will be okay. So what we need is some light. And by light here, I mean the data on the patient. Now, I will show you in a minute, or maybe I should go back for that. Yeah, let me go back to this slide. When you look at this slide, you see precision medicine for cancer is basically driven by biopsy. So you do a biopsy, and you, they take a piece of the tumor, and then they inspect this with different techniques. Pathologists do that, and then they do some genetic testing and so on. And you figure, what type of tumor it is. And depending on the tumor, you try to give the right medication. But the problem is biopsy is like a torture. It's pretty bad to go through biopsy. And some biopsies are very dangerous, especially the biopsy for the lung. It's extremely dangerous. There are people who die. So our question here is, can we get rid of biopsy to a large extent? Can we really do everything non-invasively without really doing this torture for the patient, which also costs money, by the way. So can we take an image of the patient with molecular imaging techniques, hybrid, like PET-CT, for instance, and can we derive all the information that is derived by going through this biopsy process, by not touching the patient? So I'm not touching the patient at all, okay? Just taking an image, and from the image, then we figure out if, what type of tumor it is. Is it grade 1, grade 2? Is it stage 1, stage 2? What's the prognosis? And not only that, can I also figure out the genetic composition of the tumor, like some genetic variations. How can I do this with, with an image of a PET system, which has a resolution of 3 or 4 millimeters? It's not at cell level, but we use what's called a radiomics method. That means we look at features, and those features change, the features in the image change according to the genetic composition of the tumor. So very interesting that we don't have to take any sample, we don't have to torture the patient, and we still can find out what type of genetic variation causes the cancer for that particular patient and that particular cancer. So that's kind of our goal. So we have 2 goals, actually, as I said before. One is to be able to do this only with images, non-invasively. And the second one is to use an instrument for that, which is optimized. Because instruments also have issues, as I just showed. So we have these 2 goals. Let's see what we have done so far, and we are proceeding. See. Yeah. So let's now go back to the quality issue, but in terms of precision medicine now. It's been reported that there are 250,000 to 440,000 people dying only in the United States because of medical variability. That's a large number, right? So that's been published in 2016 Johns Hopkins study. And that figure now would make medical errors the cause of 10% of all US deaths. So 1 person out of 10 in the US is dying because of medical error. And that means the 3rd leading cause of death in the United States. Trading only heart disease and chronic respiratory. So, I talked about technologies and the problem with technologies, but now I'm talking in general and the kind of problem we have. Another figure I can give is 97% of clinical trials for cancer drug, they fail. So all these patients, they get, these drugs for nothing, actually. They hope they will get better, but most of the time they don't. So we have issues that we need to try, and that's, we say that precision medicine will be the answer to that. We look at the some of the quality guys who have invented for the quality methodologies, especially Edward Deming was like the father of this movement in Japan after the Second World War. And here's a book, and in his book he explains that one of the the system of profound knowledge, that's the name of the book, if you like to read. And he says that 90% of the errors are due to a faulty system design, not human error, but system design. So we need to look at why is it we have such a big number of errors and inefficiencies, and then try to improve the system. Now, the system includes instrumentation, procedures, materials, personnel, and training management, but most of it we see it's based on instrumentation procedures. So these two are things that we need to improve. And so we look at instrumentation, we see measurement uncertainties and of diagnostic systems. Now, who knows about metrology? Have you heard of metrology? Science, metrology, anyone? Or no one? Or one person? Two? Three. Okay. Metrology is the science of measurement. And in medicine, half of the things we do is about measurement, right? Because we measure like the blood pressure, we measure temperature, we measure cholesterol levels and so on. It's all measurement. And then we make a diagnosis based on measurement. So if the measured values are not correct, if there are uncertainties, for example, if two nurses will measure your blood pressure, one will measure 120 and the other one 140, then how can the doctor make the correct diagnosis and give you the right medication, right? Now, this is for values for all sorts of measurements. So that's why calibration and so on are so important in hospitals, and we have pioneered in this, our institute. So we have this instrumentation problem, measurement uncertainties, lack of precision in therapeutic systems such as in radiotherapy, and design issues and failures such as infusion pumps that can kill sometimes if you don't, unfortunately, it don't happen. So we need actually a science that's called quality engineering and metrology. We need these two in hospital to make sure that that's actually the job of the clinical engineer and also the job of design engineers in companies like GE and so on. Procedures. So measurement protocols like data acquisition protocols for imaging systems. If you don't have the right procedure, then you don't see the tumor. Then you say you're okay. You go home, and the guy goes home, and then the next day, I don't know what happens. Same way, therapeutic procedures such as surgery, radiotherapy, drugs, all these things also need to be improved. So, if we go, let me show you some examples that we have done, such as here, we talked about instrumentation precision. That's a work that we have done with a PhD student, and that was on the design and evaluation of a breast-specific collimator using RSM and Monte Carlo simulation. That was IEEE transactions. Here we showed that we can really improve the detectability by a certain percentage, like this one. Increase of 73% in contrast line. So that means some patients now will have a more accurate diagnosis than some others. Wow. Anything counts, even if it's not 100%. Even one patient will make a big difference. So this is these are some of the optimization figures, ISO contours. Another study that we have done again with the same student, it was on improving the resolution for up to 4 times for PEM imaging, that is positron emission mammography imaging, and just by changing the algorithm. So we are doing all sorts of studies to improve all these procedures and instrumentation. Here, very important, you use PET imaging in order to find the contour of a tumor, because that contour is what you use for radiotherapy planning. Now, if your contour is wrong, so if the radiologist draws a contour which is small or large, well, that means your radiotherapy will not be effective, or it may be even dangerous. So imagine that you're, for example, giving some radiation to a non-tumor area, especially in the brain, you can kill the person, right? So we have improved by 90 times. That is 95 times less error in the segmentation with this algorithm that we have published in some journal, actually. Yeah, medical and biological engineering and computing. Here's another one. This is about mobile teleradiology performance for analyzing lung lesions. Now, when you give a therapy to the patient, you have to watch what happens every 1 month or so. You take an imaging study and then you look at the tumor if it says shrunk or not. Okay, and depending on that, you make a decision, you continue the same treatment or you change the treatment. So we make the same decisions accurately by doing teleradiology. A lot of times the doctor is not at the hospital, maybe he's in a conference or somewhere. So then can we use teleradiology with tablets and phones to do this analysis? And the answer turned out to be yes, and that was a book chapter in Spring or something. Here's another example. When we do bone age assessment, that's not cancer, but bone age assessment is an important activity when kids have some growing problems. And again, that was a study to computerize this process and make sure that we have a better precision. That's what we proved in this study. Here's another study that we have done with Florida. And here, what we've seen is hypoxia-related radiomics and immunotherapy response, a multi-cohort study of non-small cell lung cancer. Now, in this study, we found some imaging biomarkers that can tell if a patient will benefit from immunotherapy or not. Because it's only a certain percentage, like 30% of the patients will benefit, so all the rest will not benefit. So it's really a very bad idea to give this immunotherapy to everyone, right? Because they will not benefit, it's costly, and they will suffer. And they will also suffer side effects and so on. So we found with imaging, we can tell only by imaging, no touching, there's no non-invasive, if a patient will benefit from immunotherapy, yes or no. So that's something we have done with the students. This student now is in the US, and he is in a pharmaceutical company doing the same type of work for Alzheimer. Here's an interesting example that we have done. We can predict a mutation, gene mutation, for a clear cell renal cell carcinoma, so with a tumor, using CT images to see if that mutation exists or it doesn't in that particular tumor. And if it exists, then the physician has to choose a different drug. And this, again, we didn't touch the patient, we didn't do any biopsy, we didn't do any genetic analysis, but just from the images. And here is, again, we use a robust method approach for predicting the WHO ISEK grade of a RCC tumor. Again, because the grade of the tumor will tell you if it's aggressive type of when the tumor is not aggressive, then sometimes the physician decides not to do anything, just wait, see what happens. It's not necessary to operate everything. Sometimes you just let it go. Especially if the patient is very old, you know, you can afford to wait, see what happens. So that's what we did. Again, with imaging. No, sorry. I think it's the chalk or something. Anyway, here towards therapy response quantification, detecting. Okay, here we move to Alzheimer now. We also started doing the same kind of work with Alzheimer's disease. That is using, you know, precision medicine to be able to find what's wrong with the patient. And here, especially, what we've done is try to measure the severity of the, well, making a diagnosis, making a diagnosis, which is early diagnosis, but second, to determine the severity of the disease, of Alzheimer's disease, in a accurate way, accurate and precise way. This is again a new thing because so far people used to do that. By using psychoneurological tests. So cognitive tests, you know, you take a test, you know, do you remember this, you remember that, so there are some questions as different types of tests. So then those are more very subjective and they're based on symptoms. But PETs can actually, this molecular imaging PET systems, they can show you that there is a beginning of Alzheimer much before the symptoms occur because they show the biology. Okay. And anatomic changes occur much later. So,
They use amyloid for them.
yeah. Okay, so that was an example we have done and we published that. Now we have published the second one. We show that actually, this index that we found is proportional to the results of the psychoneurological test. So that means instead of doing a subjective test, we can do a scan, again, non-invasive, but it's very, very accurate. So we can tell, maybe we'll see that, we don't know yet, but maybe within 2 weeks or 3 weeks, if the patient has gotten worse or better. Now there are some new drugs that came out for Alzheimer's disease, so we hope we can move towards therapy, not just watching the degeneration, but maybe we'll see now an improvement in Alzheimer's. So that's what we're doing, again, with precision medicine, but this time regarding Alzheimer's. So where do we stand? We see that we have lots of work. I mean, you touch any subject in biomedical field, and that subject will need tremendous work. So that's a field without any limits. And the glass is half empty. So it's not half full, it's not completely empty, but it's half full, half empty. So we need to keep being optimistic and work on these topics. And hopefully one day we'll arrive to 100% efficacy, for serious diseases such as cancer. So the current work we have now involves oncology, but also some neurology like Alzheimer's disease, and especially trying to move into the omics area. So get more into the genetics and other proteomics and other omics area to see if we can also touch those and improve accuracy. For this, we have 2 courses. One is on nuclear medicine instrumentation, which is PET and Spec, is molecular imaging modalities. And the second one is quality assurance for biomedical engineering, which shows how to improve systems, what type of optimization method exists for improving the precision of the systems. So that will be it. I don't know how much time I took, but So I thank you, and if you want to reach me, I have a page, and can reach me on that page. If you have questions, and I can try to answer. Where is James Hocam? Oh,
Go ahead, Hocam, for questions, you can get questions from the audience or here.
okay, you can moderate. Hello.
Please go ahead. Are there any questions? Did you say? There was a technical problem, so I had to step outside to solve it, but is there any? So precision medicine is the future.
Well, we try to
Go ahead.
reach 100%. I mean, that's, yeah.
Go ahead.
I'm just wondering, do the methods to determine a precise treatment require individualized techniques?
Do we have a what? Methods for?
Like determining which personalized technique to use also requires specialized.
Okay.
I was wondering,
Yeah, there are. We're not doing this though. I mean, if you're talking about drugs, there is another area which is called drug design. So trying to find different targets for drugs and develop this drug by using computerized methods. Actually, there is a guy in New York. He has a big company on this. He's a big shot at this moment working on this. But many people are also working on this issue. There is also brachytherapy, actually, people in RT trying to improve the RT therapeutic efficacy of RT, like motion control and yeah, that's a big issue, and also real-time control of motion and segmentation, which is also something we tackled. So there are different aspects that everybody is doing something. Oh, yeah.
You mentioned segmentation, it has prospects of yielding higher accuracy in terms of earlier diagnosis for cancer.
Earlier diagnosis of cancer, it looks like the road to go today is liquid biopsy. So that means taking the blood sample and then from the blood, trying to figure out some biomarkers that show that there is cancer. And then the next thing you do is imaging. But then again, when you do your imaging, as you could see, you will still miss some of the small tumors, you know. So that's what we have been trying to do, actually, also with others, trying to improve the resolution of the PET system, trying to improve the detectability, improving the protocol and so on, so that you don't miss. But probably the early diagnosis, because you don't send someone to a PET scan if there is no reason. It's expensive, and you get some radiation and so on. So, but if you can get this tip that there is something, some suspicion from the blood, as we call, you know, liquid biopsy, then that's probably the future. Any other? Yeah, it's perfect.
Thanks.
Okay. All right. One more.
Sorry, just a general question. The way I understood precision medicine means that you're having very individualistic approaches to diagnosing and treating a patient. Do you think that it's viable in the long term, because there's so many costs involved and complications? You're going to have a specific drug for each kind of different patient, for treating or for.
I don't think that's going to be the big cost. I mean, today, for example, if you take one of the most interesting things today is probably gene therapy. You know, I'm probably molecular biology guys here, maybe. Who is a molecular biologist? Any or there is a thing? You? You are molecular biologist.
I my
Okay. So Yeah, I mean, there are this kind of new techniques, and there are, from what I heard, 200 new drugs coming up every year by this company. The FDA is trying to, check them out and give them permission. But there's quite a lot of work in this direction. But that's the only way. I mean, because the other way is not working. That is giving chemotherapy and radiotherapy the same, pair to every patient the same way. Doesn't work. It doesn't work. We have like 20% efficiency overall. So that doesn't work. So the only other approach is to find the driver gene mutations and maybe also other organ stuff, and then try to find the right drug. And what we are doing is, non-invasively trying to map the biopsy results by using only, which is a non-invasive way. So torturing the guy and putting some needles and pulling out some stuff from the bio. Doesn't look very good.
Right.
That's it for me.
Okay, are there any further questions? We still have 3 more minutes for questions from the audience. I don't see any audience from here. If there are YouTube watchers, I don't know, they should be monitoring.
Okay.
Our assistant should be monitoring. If there are YouTube watchers, they can ask them. We can maybe stop the recording now. And I thank you, Alberto Hocam and Professor Güvenç for that. With that, recording session, if I have to record it here, let me see. Yes. And then informal olarak olmak istediğim bir şey varsa hocamız burada konuşmayla ilgili. Bir de ufak kültürümüz var. Daha önce mamaları yediniz ve sandviç yiyenler veya sandviçlere yeni ulaşanlar varsa çıkışta hemen sol tarafta biraz içecek, biraz kurabiye ve gazlı sandviç. Orada da sohbetimize devam ederiz.
Teşekkür ederim.
Sağ olun. Hemen bakın sol tarafta. Hala orada öğrenciler bıraktık. Sandviçler var.
Evet, yardım
Yardım sıkıntı yok. Yardım gerekli. Genel değerlendirmeleri geçirmiyoruz. Direkt çaktırıyoruz. Genelde çaktırma eğiliminde oluyoruz yani. Sonra geliştirince öyle
Şimdi protokol için genişletmem gerek.
evet, odaklanma. Biraz da şeyle konuştuk olurlar. Seni gruba katacağım. Onun da kafasında benimle ilgili bir şey varmış. Osman Hoca vardı ya. Sen bir Osman Hoca'yı ziyaret et olmadı. Oradaki dokümanı al. Çünkü Osman Hoca'nın konuştuğu muhabbeti geçti. Bir takım yerlerde ondan geçti. Osman Hoca'yı dünya kadar güzel kıl. Yani hocam çay kahve lazım. Ne yapalım burada gelirim ben istediğim kadar falan gibi. Onu yakalayıp imzasını alalım. Dokümanı da alacağım. Hoş geldin. Sonra da sonra da onları ben katacağım. Uzayla bir görüşeceğim. Evet. Sonra ilerleyeceğim. Şu arkadaş bu genel durumu nasıl? Toparlamak kolay değil. Ne? Böyle ne yapacağız? Ne yapacağız? Ya da hemen hani de devireceğim yanına gömedim. Hani ya o tarafa geçeceğim ya bu tarafta kalacağım. Anlatıyoruz. İki dolmaya geldik. Arkadaşa bir çalışacak masal, sen startup firma kuruyorsan aynı masa kiralayabiliyorsun. Yoksa öğrenci kontenjanından TEDO tarafından ücretsiz de değil. Onların da bir parası var. Kuluçkaya gelebiliyorsun, oraya bir gel. Sonra da bizim tarafta nükleerde ne oluyor, ne bitiyor? Bir dahil ol. Sonra da projenin takip aldıktan sonra bazı işleri, belki gelişimleri ilgili. Nükleer tarafta bir şeyler geliştirelim. Ben şeyle konuştum. Erkçen'le konuştum bu arada. Önümüzdeki Cuma'da da geliyor.